and it will be posted on our website if you'd like to watch it later. So just a moment before we kick off, please. Okay, one more person joining. Okay, well, time to kick it off. Thank you again welcome to the Nyrada Investor Briefing for today. Thank you for joining. My name is Dimitri Berstein. I look after investor relations. With us also is James Bonner, our CEO. This webinar, as mentioned, is being recorded and will be posted on our YouTube page soon. The reason we're having this webinar is that it's been a fairly big year for Nyrada and a fairly big year to come. James will go into the details of that. It's been, as mentioned, a significant and transformational year. Before we move forward, just a couple of housekeeping things. Of course, we have to mention our, the mandatory disclosures about the information here is not advice, and be careful what you read. This slide deck has also been released to the market this morning, so it's available to review at your pleasure. We take the opportunity to welcome any new investors who've joined since our last webinar, and just to remind anyone online that we have an SPP that's currently open and will be closing on Monday next week. So please take the opportunity to have a look at the information booklet. Okay, I'll hand over to. Before we continue, the format for today is not going to be James running through a set of slides because they've been released to the market. What we're going to do is I'm going to ask James some of the most common questions we're asked. Hopefully that'll address any queries that you have. If there's any questions you have that haven't been addressed, please put them in the chat box and we'll try to ask them during the presentation. We have to close this at 9:45, so with no further ado, I'll hand it over to James.
Yeah, thanks, Dimitri, and just want to extend a personal welcome and thanks to everyone who's joining this webinar this morning. So, as Dimitri said, this year has been a very busy period for the company, and we've had a lot of success. So, I'll just start by running through the list of key achievements for the past 12 months. So, back in February, we released data from a stroke study. This was a mouse model of ischemic stroke, where we saw an impressive efficacy result, a 42% neuroprotection. Second to that, we also released data more recently in October. This is a rat study that we ran looking at myocardial infarction or coronary heart attack in a rat. And we found that the drug also conveyed significant cardioprotection. So we're going to talk about those results in a little bit more detail and lay out what the plans are for the next 12 months. The other significant advancement was the recent completion of a battery of safety and toxicology studies, and these were completed in rat and dog. And these are a comprehensive set of studies that are required to be run for a novel new drug before it can go into a first-in-human study. So we'll talk a little bit more about that as well. Also ongoing is a study that we're running in collaboration with the Walter Reed Army Institute of Research. This is a DoD or US Department of Defense branch that specifically looks at traumatic brain injury. So we've had a long-standing collaboration with them. We've got results that are coming out in January. So we're happy to take some questions on that as well. The final achievement, I guess, is the advancement or imminent advancement of the drug NYRBIO3 into a first-in-human phase 1 study. So we'll talk a little bit more about that as well and take any questions.
Thanks, James. So at no further ado, let me put the first question to you, James. What is the problem NYRBIO3 is looking to solve?
So NYRBIO3, our lead molecule, has been developed with a very specific role in mind. It's an ion channel blocker, and it's a drug that works on a novel target that's only fairly recently been studied. And we see it as a potential first-in-class treatment for stroke, for traumatic brain injury, or TBI we'll refer it to, and also coronary heart attack. So all of these events are serious and life-threatening, and we're excited by this molecule, given the efficacy data we've seen in preclinical studies, with it showing not only neuroprotection but also cardioprotection. So we'll talk a little bit more about how that works. The drug itself has been developed as an acute treatment, and it would be delivered to patients in an emergency setting by intravenous infusion. So in terms of the big picture opportunity, we see NYRBIO3 is representing a transformative solution for these critical medical conditions. And you'll see as we go through this that for some of these indications or events we're talking about, the current treatments are either limited or don't exist, and it's certainly in the case of traumatic brain injury. So we're talking about very large, and we see them as underserved markets. And if you look at the slide that Dimitri shared there, there's the potential for up to $60 billion US dollars in market potential across these three indications.
Thanks.
I guess just one final point, and sort of just to summarize what's on the slide. We see this as a one drug, two applications, three major markets.
Thanks, James. The company's just raised 3.3 million in capital with an SPP currently running. What will this capital be used for, and how long will it last?
So the capital that we've just raised will take us through the phase 1A, and we'll talk a little bit more about the phase 1 study in a later slide. But that's a key first-in-human milestone to confirm safety and tolerability of the drug, and also to determine pharmacokinetics, which is essentially drug levels in the blood following administration. The money that we've raised will also cover some R&D that we've got planned, looking at other indications where this biological target, these TRPC ion channels, are relevant. I'll talk a little bit more about that as well. Dimitri mentioned there's a share purchase plan that's open currently. We're looking to raise an additional $1 million Australian dollars in that share purchase plan, and that will, I'm confident, take us into the second part of the phase 1. This is where we dose patients or subjects for longer.
Okay. What is the status of the phase 1 trial, James?
So the phase 1 trial is in the late planning stages. We're just assembling the documentation required to submit to the Human Research Ethics Committee, and so we're engaging with a site and a CRO, a contract research organisation, to compile that documentation, and it's essentially distilling down what we learned in the safety and toxicology rat and dog studies, and putting that into an investigator's brochure, which describes everything we know about the drug, and also drafting a clinical study protocol. They're the main documents, and that's well advanced. We're looking to get the ethics committee application in imminently. And once that's approved, that will open the way for dosing the first cohort of patients. In terms of how long the phase 1 study will run, we have, as you see in the graphic here, initially 3 cohorts. Each cohort we estimate will take approximately 1 month. The first part of the study is dosing with a short duration infusion, 3 hours. The second part of the study will be dosing up to 72 hours. So each of those cohorts will we think take approximately a month. So that takes us into to the middle of calendar year 2025. And one thing we'll be doing as we work through the phase 1 study is we'll be communicating the results of each cohort to the market in an announcement. Just in terms of the study design, this is a randomized placebo-controlled double-blind study. So what that means is that some of the subjects, and these are healthy volunteers we're talking about here, some of them will receive the drug and some of them will receive placebo. But the subjects and the study staff, and indeed the staff at Nyrada, they won't know what treatment is assigned to what subject. So that's kind of where we are in terms of the study itself and what it's designed to show. It's primarily a safety and tolerability study. So we'll see how the drug is tolerated by healthy volunteers, and it will enable us to pick a dose and a dose regimen for phase 2 studies that will follow.
And speaking of phase 2, James, what might a phase 2 study look like?
So a phase 2 study, that's a study that's conducted in the target patient population. So we'll be studying the drug in either ischemic stroke, traumatic brain injury, or coronary heart attack. And these studies typically enroll, say, up to 100 patients. And with the cardio study, the coronary heart attack study, we're looking to run that here in Australia. Similarly, the stroke study. With the traumatic brain injury study, we would love to have some sites here in Australia, but we really need access to patients, and that drives recruitment rate and the duration of the study. So we see that as being a study that we would primarily focus in the US.
What is the study, moving on back a little bit, what is the status of the Walter Reed study? And if you can just describe what it is, please.
Okay, so the Walter Reed study is a study of the drug in a model of traumatic brain injury. And it's in a model of traumatic brain injury that's specifically relevant, or particularly relevant, to the military. So in this study, we're looking at penetrating traumatic brain injury. So this is the sort of injury that you would sustain following, say, a bullet wound or a shrapnel wound. And it's obviously a high priority for the military for obvious reasons. We've done an extensive pilot work with this model, and we've just run the in-life phase of the drug study. And this is a collaboration with Walter Reed and also UNSW here in Sydney. And the samples have been scanned by MRI at UNSW, and the data analysis is ongoing. So as we said, that the study is due to read out in January.
Okay, one of the key things for a drug development company, obviously, is patents and patent protection. What's the status of BIO3's patent?
So NYR-BIO3 is a novel molecule that our chemists have developed. And so we've lodged a patent, provisional patent for that. And what that patent covers, it's called a composition of matter patent. So basically it's protection of the molecule plus closely related analogues. And we've also included in that provisional patent the indications that we intend to go after. So in terms of patent protection, a composition of matter patent offers a very high level of protection from other competitors. And it's preferable to, say, a method of use patent or a formulation patent. The provisional patent will be published in 12 months' time, and it will give us, from the filing date, which was in October, 20 years of patent protection once granted. So that's a typical life of a patent for a drug. And in terms of the territories that we'll look to get coverage, the obvious one is the US, because it's such a huge market, and also Europe and the countries that make up the EU, and Japan. But we're also looking to get protection in countries with large populations like India and China.
Thanks, James. Non-dilutive funding is often an important source of funding capital for small biotechs. What's the opportunities before Nyrada?
Well, they're many and varied. With traumatic brain injury, there are non-dilutive funding opportunities available through the US Department of Defense, and we keep a watching brief on those opportunities. And it's really a matter of timing for the right grant opportunity to come along at the right time. The DoD grants, they tend to cover not only preclinical development but also clinical development as well. And we see ourselves as well placed given the long-standing collaboration we've had with Walter Reed. Outside of that, there are lots of other government programs, and not only in the US through the NIH, but also here in Australia through some of the governing bodies here. And just recently, for example, there was a Royal Commission report published, which made a recommendation to establish a brain injury program. So we're interested in that. And outside of that, the Department of Health also regularly publish grant opportunities that we hope to take advantage of.
James, just on that one, for those on the line who aren't aware, James had well, I won't say a similar role, but came from Neuron where he helped develop their brain injury program, and Neuron was successful in gaining some capital from the US DoD. Obviously, some of your experiences could be of value to Nyrada on that.
Yeah, so anyone who is invested in or follows Neuron will know of the drug Daybue or trofinetide. Interestingly, that started out as a drug to treat traumatic brain injury. So I oversaw the traumatic brain injury program, and as Dimitri mentioned, that was funded in large part by the US Department of Defense. So I've sort of been down this road before, and looking forward to getting into the clinic.
Great. And speaking of commercialized brain injury programs, how long do you think before NYR-BIO3 may be commercially available? What's the clinical pathway for any of the indications you've indicated?
Yeah, so the phase 1 study that we're running has been designed to support phase 2 in either stroke, traumatic brain injury, or coronary heart attack. Now, the phase 1 study, which I've talked a little bit about, that is anticipated, or typically phase 1 studies run over about a year. But drug development runs in 3 stages. And the second stage, beyond testing the drug in healthy volunteers, as we're about to do, is phase 2. And phase 2 studies are in the target patient population. They're typically larger in scope, and phase 2 studies typically run over about 3 to 4 years. Phase 3, which is the final stage before approval, they tend to run over 3 to 5 years. But there are a number of things that drive the timing, particularly for phase 3. For example, do you have to do a single phase 3, or do you have to do multiple phase 3s? Also, we plan to run these programs under the auspices of the US Food and Drug Administration, and they offer expedited pathways that we're going to look to take advantage of. And examples of those is fast track designation, breakthrough therapy. So these are all options that are open to us with NYR-BIO3. But the length of time largely depends on a number of factors. And another one is the therapeutic area. If you're developing a drug for an orphan indication where only a few patients are affected, you get accelerated opportunities for development. But of course, along the way, there can be failures and delays. And one of the big drivers of completing these studies is the time it takes to recruit patients. So a lot of factors.
As you said at the beginning, this past year has been fairly important, and this coming year will be even more important. So what do you see are the key milestones for Nyrada for the coming 6 to 12 months?
Well, the key one is the start of the phase 1 study, and the updates we'll be putting out around that. The readout from the Walter Reed study, which is due in January. We'll also be lodging IND submissions to the US FDA to support phase 2 development. And we're actively pursuing non-dilutive funding opportunities. So where we're successful, we'll certainly announce that to the market.
Just quickly, just for those on the line who may not be familiar, what is an IND, and what is the difference between drug development in Australia versus the US?
Okay, so an IND is an investigational new drug application, and it's a requirement before you can dose a patient or a subject in the US. So in Australia, we have a slightly different system. It's managed under the Therapeutic Goods Agency, the TGA, but a lot of the review and approval is decentralized to human research ethics committees. So an IND is mandatory before we can study the drug in patients in the US.
Is Nyrada looking to partner with a pharma company, and is there any interest in these indications or in this space and the work Nyrada's doing?
Well, that's an interesting one. I think there's always been an interest in neurology drugs, particularly first-in-class drugs, and particularly where those conditions are serious and life-threatening. We expect the cardiac indication to be out-licensed at some point, possibly before phase 3. One of the reasons for that is that with a cardiovascular drug, the size and scope of the studies is generally very large. It's a high capital input exercise. I think with stroke and TBI, there's an opportunity, particularly for TBI, and if we can attract non-dilutive funding, to take that all the way through to registration. But the focus of the company at the moment is really just to build value in the asset. My chairman told me that good assets are bought, not sold. So focus on getting through phase 1 and then phase 2. And I'm sure if we're successful in doing that, there'll be no shortage of interest.
On a similar theme, whilst obviously the entirety of NYRBI3 can be out-licensed, it's possible for the company to out-license it indication by indication, isn't it?
Absolutely, yeah. And they're quite different indications, and have to be treated differently. And as I said before, with the development of a drug for traumatic brain injury, I think there's definitely an opportunity there to take advantage of expedited pathways. And if you look at severe traumatic brain injury, that's classified or could meet the criteria of an orphan drug indication. So these are all things that have to be factored in.
Okay, so perhaps we now move on to the actual nitty-gritty of the drug, and to ask some of the more science-based questions. How does NYRBI03 work?
Okay, so Dimitri's just shared a very busy slide, and my background's in chemistry. I'm not a cell biologist, but I'll try and talk through this in simple terms. So the drug has been developed with a very specific role in mind. And if we take a step back, I mean, the genesis of this work this program started with the development of a knockout mouse where the target that we're going after was genetically knocked out. Now, these mice, they have a normal lifespan, they're breeding capable, they suffer no adverse effects from having these ion channels knocked out. But what was remarkable about these knockout mice is that they were neuroprotected in models of stroke and traumatic brain injury. And so some pioneering work was done at UNSW here in Sydney, looking at these knockout mice in a model of ischemic stroke, and that was proven to be the case. So the job of the chemists at Nyrada was to develop a molecule that essentially blocks this channel, effectively doing the same job as knocking it out completely. So if we look at the image in this slide, I won't go into too much detail, but you can see the trypsin channels, the target of the drug, are the pinkish salmony-coloured tubes or columns on the right-hand side. Now, this image represents the surface of a cell, and it's generic. It could be any cell. In the brain, it could be a neuron. In the heart, it could be a cardiomyocyte, which is the cells that make up the heart muscle. And what we're effectively doing is blocking calcium entry through these ion channels. And what sets that off is an agonist, and you can see that on the left-hand side. And this agonist, and in the example of a neuron, would be glutamate. In a cardiomyocyte, it could be angiotensin. And what these agonists do is that they bind to their receptors like a key going into a lock, and they set off a biochemical cascade. And what that does is it keeps these ion channels in the predominantly open conformation, and that lets this calcium into the cell. And what that does is it can cause a whole cascade of effects leading to cell death, neuroinflammation, and so on. So in the table there, you can see the cell types that are listed that express these trypsin channels. And in the brain, you've got neurons. I think most people know what a neuron is. And blocking the ion channels stops activation of these channels, and it reduces neuronal loss. Astrocytes, they're like the helper cells in the brain, and they play a key role in cleaning up the mess after an injury. And trypsin channels are expressed on these astrocytes, and activation of these channels can lead to swelling. And microglia, they're the immune cells in the brain, and also they get activated with harmful consequences. They basically trigger an immune response in the brain that gets out of control. In the heart, the cell types we're talking about, the main one is the cardiomyocytes, which make up the cardiac or the heart muscle. And if they get overactivated, that can lead to damage. So that's kind of like the long and the short of it. So we're basically blocking that activation of these ion channels, and that has a positive therapeutic effect. Okay, so what does that look like in terms of neuroprotection? And here we're talking about stroke and traumatic brain injury. You can see on the left-hand side there, there's a blue and a green panel, and these are rat brains that were taken from the pilot study we did with the Walter Reed, and you can see there's a significant injury there. This is a penetrating brain injury, and in the hours and days that follow that primary injury, you can see that it expands. If you look at the green image at the bottom, you can see that there's quite a significant expansion of that injury. So what the drug is designed to do through blocking activation of these trypsin channels is to suppress that secondary brain injury. And what that means for the patient is that they have a better chance of surviving the injury, but they also have a better prognosis in terms of their recovery and level of disability, and you can see that sort of graphed to the right there. So with a treatment for traumatic brain injury, we're not talking about a silver bullet cure here. We're talking about a drug that prevents or limits the damage following the event. And if you speak with clinicians, they talk about even a 10% improvement on one of these outcome measures could mean the difference between a patient being able to drive or not, being able to live alone. So, even a small improvement can lead to quite a profound effect on the patient. Next one, Dimitri. So, in terms of cardioprotection, and this is sort of something we got onto fairly recently, and it was following research that was published in the TRPC knockout model that I mentioned before. So we tried our drug in this rat model of myocardial infarction, reperfusion injury, or coronary heart attack, and you can see we got quite a profound result. We got an 86% cardioprotection with the drug, and that's protection of the cells that make up the heart muscle. In addition to that, we also got significant functional improvements as well, which you would expect to see. We got a 43% increase in left ventricular ejection fraction, and that's the amount of blood the heart can pump with each beat. So that's a measure of how the heart is performing, and also we got a 50% increase in fractional shortening, and that's a measure of how well the heart is contracting. In addition to that, we also got really impressive reductions in blood injury biomarkers. So taken together, it's very strong evidence of cardioprotection, and it sort of mimics what was found in the TRPC knockout model. So we're very excited about this data, and it opens up a new avenue for the molecule.
Thanks, James. You've spoken mainly about 3 potential indications. Is there any opportunity for other indications where TRPC channels play a role?
Well, yeah. So calcium dysregulation plays a role in a number of different diseases, and these TRPC channels are also expressed in a number of different cell types outside of the brain and the heart. For example, in the kidneys. Blocking TRPC channels is thought to prevent renal fibrosis, and also TRPC channel signaling plays a key role in pain. So these are 2 other indications that we're looking to study with the drug. One thing we're also doing is developing an oral form of NYRBIO3. Currently, it's an intravenous dose form. And yeah, we're looking at a drug that can be taken in pill format, which would be more applicable to these other indications.
Thanks, James. That's probably all the main questions the company tends to receive. Now's probably an opportune time to throw the mic open to anybody on the line. If you have any questions, please put them into the chat or put your hand up, and we can, open your microphone to allow you to ask yourself. We don't have any on-hold music, so
everybody,
if you have any questions, please.
On Monday morning.
Yeah.
I hope I haven't put everyone to sleep.
It doesn't appear to be any further questions, so unless my system's not telling me, that appears to be it. James, is there any closing comments you'd like to make?
Yeah, why don't you advance to the conclusion slide?
Oh yeah, good point.
Yeah, so I guess just to summarise, we're excited by NYRBIO3. We see it as an innovative therapeutic, and it's designed to act on a novel therapeutic target that has a very solid scientific basis. We've got a strong pathway to clinical trials and to commercialisation, and we've got some exciting milestones on the near-term horizon. particularly as we go into phase 1. We've got potentially more, beyond the Walter Reed readout, potentially more efficacy readouts down the track as we look at other indications where this drug might be useful. So I think we're making strong progress. It's been a good year for the company. We're well placed to advance it through this key milestone. And I would encourage you to take another look at the share purchase plan document and join us on the journey. There's a slide in the deck that has a QR code that will take you to the website and allow you to sort of take a deeper dive into the opportunity. And if you want to know a little bit more about the different indications we're pursuing, there are some other slides in the deck that was lodged today with ASX that should help answer some of those questions.
One late question has come in from the floor, James, and I'll read it verbatim. What is the difference between Argenica and Nyrada?
Oh, okay. It's very difficult for me to make any commentary on other drug programs because this is a complex business and there's quite a lot to it. I mean, I have read a little bit about what Argenica are doing. They're developing a peptide. I guess that's one of the key differences. We're working with a small molecule, an MYR-BIO3, it's going to be interesting to see how that development program goes.
Thanks, James. Last call. That appears to be it. I'm just checking for hands up. Nope. Thank you all for your attention. Again, as mentioned earlier, this presentation has been recorded. It will be posted on our YouTube channel and made available later today. We appreciate your participation. We appreciate your support. For those who are eligible, we again invite you to have a look at the SPP information booklet. And if you have any other questions, please feel free to reach out to James or myself. We can be both reached at info@nyrada.com. And at this point, we'd like to thank you again for your participation. Have a great day and onwards and upwards.
Thanks, everyone.
Thank you.